Now if you read the part 4.1 of question 4 which asks complete the sentences. First part asks you the tissue with the most chloroplasts, and the answer is palisade mesophyll. Secondly, the tissue with many air spaces, spongy mesophyll. Thirdly, the tissue that differentiates throughout life, meristem. Let's move on to next question part 4.2 that asks you to name material what strengthens xylem besides cellulose. And the answer is, and the best answer is lignin. Let me give you an example tip. The meshwork of fibrous network of cell wall is composed of strengthening materials such as cellulose, hemicellulose and lignin. Easy memory trick. Lignin equals line that keeps plant in line, upright. Let's read the next question part 4.3 which asks you to process transporting dissolved sugars, and the answer is translocation. Next question requires your deeper understanding about active transport, and the AQA asks you command words such as explain the processes, so the question is 5-mark explanation. Part of the question here on slide is 4.55 marks here. Sugars move by active transport. Active transport requires energy. Energy is released during respiration. Mitochondria in companion cells provide ATP. Carrier proteins in the membrane pump sugars against a concentration gradient. Notice how each sentence adds a scientific detail. That's how you build 5-mark answers. 4.6 asks you to answer the change happening when undifferentiated cells become phloem cells, so examine requires your understanding of command word where he asks you to describe one structural change in phloem. Note it's a 1-mark question, hence instead of long explanation one fact would be enough. So the answer would be that end walls break down to form sieve plates, or the cell loses most of its organelles to allow more space for flow. Will award mark. Here is the exam top tip. Don't confuse. Transpiration is water in xylem. Translocation is the sugar in phloem. Remember, phloem transports food. Let's move on to the next slide where students investigate the effects of salt concentration solution on uncooked potatoes pieces. Hence the question 5 carried out osmosis investigation, not only the definition but also applying the understanding of the id scientifically. First question starts with the experiment setup. The part 5.1 of question which is 6-mark method question. Step 1. Cut 6 potato cylinders to equal size. Step 2. Dry and record initial mass. Step 3. Add different salt concentrations to labeled tubes. Step 4. Place 1 cylinder in each tube. Step 5. Leave for a fixed time. Step 6. Remove, dry, record final mass. Then calculate change and plot a graph. Top tip. For 6-mark methods always say, repeat the experiment. Calculate a mean. Control variables temperature, surface area, volume. That alone pushes you toward full marks. 5.2 Explain the result for the potato pieces in the 0.6 mol decimeter cube salt concentration. 3 marks. According to the graph in figure